In this video, we'll discuss the structure and function of ribonucleic acid. RNA, like DNA, has a primary structure consisting of a sugar phosphate backbone that is formed by phosphodiester linkages, and extending from that backbone is a sequence of 4 types of nitrogenous bases. The primary structure of RNA differs from DNA in that RNA contains uracil instead of thymine as one of the pyrimidines, and it contains ribose instead of deoxyribose as its sugar. The presence of a hydroxyl group on ribose makes RNA much more chemically reactive than DNA and also at the same time less stable than DNA. The secondary structure of RNA results from complementarity between base pairs. Bases of RNA typically form hydrogen bonds with complementary bases on the same strand. RNA is a single-stranded molecule, so bases on the same strand that are complementary to each other interact and form hydrogen bonds. The RNA strand folds over, forming a hairpin structure. The bases are on one side of the fold, and they align with an antiparallel RNA segment on the other side of the fold. You can see the hairpin structure of RNA in this image. It's a single-stranded molecule that forms a loop, resulting in the formation of hydrogen bonds between complementary bases on antiparallel segments of this loop. In addition to a secondary structure, RNA molecules can also have a tertiary structure when the secondary structures fold into more complex shapes. Just like DNA, RNA can store information and is capable of self-replication. Structurally and chemically, RNA is intermediate between the complexity possessed by proteins and the simplicity of DNA. Let's compare the structure of DNA to the structure of RNA. The primary structure of both DNA and RNA consists of a sequence of nucleotides, the difference being that RNA contains uracil instead of thymine, and the sugar in RNA is ribose, whereas the sugar in the DNA backbone is deoxyribose. The secondary structure of DNA consists of 2 strands wound in a DNA double helix, whereas the secondary structure of RNA is the same strand that loops around to fold upon itself. At the tertiary level, DNA doesn't have a tertiary structure, but RNA can fold to form distinctive 3-dimensional shapes, as can be seen in this tRNA molecule. RNA can also self-replicate. This process is slightly different from DNA replication. During RNA replication, 2 successive copies of the template strand are made to get a strand that is an identical copy of the original template strand. Let's look at an image of this process. Nucleotides are added on that are complementary to those on the template strand. This growing nucleotide chain polymerizes and separates from the template strand. This copy now serves as a new template to generate another copy that polymerizes to form a strand that is identical to the original template. So if you look at the original template strand and the second copy that is made, they're identical to each other. RNA is a very versatile molecule. It can function in catalysis. For a long time, it was thought that RNA didn't have any catalytic activity, but several ribozymes, meaning RNA molecules that have catalytic activity, have been discovered. Because RNA has a degree of structural and chemical complexity, it is capable of catalyzing multiple chemical reactions. Because RNA can undergo many levels of folding, it is a more versatile molecule than DNA. An example of such a structure is tRNA, which is an adapter molecule during protein synthesis. Another example of RNA molecules that are part of a complex catalytic machinery are the ribosomal RNA molecules that help in synthesizing proteins. And a third example would be messenger RNA, which is also involved in protein synthesis. This concludes our video on RNA. At the end of this section, I'd like you to be able to answer the questions that are listed on this slide. Thank you.